Yeah, so thank you for the introduction. So I'm Esben Bjerrum, and I work as principal scientist at AstraZeneca, where we work with deep learning for various drug discovery tasks. But I'm going to talk more about this. So a quick agenda. I know not everybody here worked daily with chemical modeling and machine learning for chemistry. So I'm going to make a very brief introduction how to look at how we could model molecules, and also look at some of the data augmentation strategies that we can use to improve our modeling. And then I go to answer some of the questions where we have we make de novo design. We design molecules. We get the computer to design the molecules that we want for us. And I will talk a little bit about some of the ways we can do that. And then I will also, in the second half, I will look a bit on reaction prediction, because we also want to be able to figure out if we can make them compounds, and also give a suggestion for how. And then in the last part, I'm going to look a bit about how we can actually use the same model to do both tasks. First, a little about AstraZeneca. I mean, we are a global company, and the Gothenburg site here is one of three main research centers we have. So we are a lot of scientists here working with a lot of the aspects of drug discovery and development. Because I work in drug discovery, but that is just the beginning. It takes a long time and costs a lot of resources to do the research and development necessary for launching a new medicine against untreatable disease. So first, we must, of course, find the potential molecules that we want to do, and then it must be developed into a drug that can be administered to the patient, and it needs to be tested. And finally, we can send the approval, the documentation for the authority is safe enough, and we believe it works. We can see that from the clinical trials. And then we can finally get to launch the medicine, and we also need to do some post work there and actually see how it's used. Is there anything that comes up there? And then, of course, patents also run out, and then you also need to see that there come generic parts. So how do we do in drug discovery? Usually we have a starting place, right? We have identified a hit compound. It can be found via high throughput screening or DNA encoded libraries, or maybe from some knowledge about natural compounds that have an affinity for this protein, or it can be a academic research finding or something. But they are often not drug-like. They may be weakly active. They may be unselective. There could be a toxicity risk we don't want to have. They could be not very stable, and so forth. So we need to optimize these, and we do that via design, make, and test, analyze cycles. So we design new compound ideas. We make them in the laboratory. We test the properties of it. We analyze that. Did it improve? Did it not improve? What is a new understanding of maybe the structure-activity relationship in the binding pocket? We design new ideas, and through that kind of iterative process, we end up with a compound that is a candidate drug. So we want it highly potent, and it should be effective. It should be more or less have the right stability, and we should have no fear of any toxicity issues. And then we can say, okay, now it's time to get it to development and take an investment decision to actually turn it into a drug. So our challenge in my department is to find ways that we can speed up and improve this process using AI. So just to dive a little into the chemistry parts, is that one thing that have been around for some time is quantitative structure-activity relationship. or QSAR modeling. It was machine learning before that became a buzzword, but done on chemical data. And of course, there have been a long development of what models we use and how we create, how we represent the molecules for these models. So there have been a lot of engineered features, various descriptors. You can also use simple descriptors here, molecular weights, hydrogen bonding donors, maybe calculated properties. And there's also a lot of ways that we can create a fingerprint where we may use different substructures, is the substructure present or not present. And it can also be a more systematic analysis of the computational graph. So we take our moles, we turn them into some kind of features, and we have our model, and then we create, we can model that towards the response that we're interested in the property. And one of the formats we have been looking at to do deep learning on, where we actually not featureize things, is SMILES, which is a simplified molecular input line entry system. So it's a way to take the molecular graph and turn it into a string notation, a linear notation. And the way we do this is we have brackets. So if you reach a branch in the molecule, you can say now we branch off, and when we close the bracket, we go back to where we were before and continue. Or we can also use numbers to say here's an extra bond between areas in the graph. So that's a very convenient format if you want to send molecules in plain text and emails, or store them in a spreadsheet or something. But it actually have also turned out that it's very efficient when we want to use natural processing architectures that have been developed in the recent years. So one of the ways we can do that is via recurrent neural networks. We have had great success with long short-term memory cells that has a little internal state inside the cell that's get propagated through the inputs. So first you input one character of the smile, and then you change the internal states, and these states are then progressed to the next one, and you input the next one, and the states can be changed. And in the end, you can look at the states or the output from the model, and through a small feed-forward neural network, you can model the property that you want. So this is actually a way to avoid the featureization, and then the LSTM network will learn what kind of properties in the SMILES is important for the prediction. It works surprisingly well, but we can also do a little data augmentation trick. If you have looked at image recognition, they also use a lot of data augmentation. You have only a limited set of labeled images, and you want, of course, to expand this. So you can take a picture of a cat. It says cat sitting in the forest, but then we can do various permutations to this image. We can mirror it. We can maybe change the colors of it. We can crop it in various ways, or maybe we can even turn it upside down or rotate it some way. So this is a way it has long been known in image recognition, that this increase the robustness of the final network. And in SMILES, we can do the same thing because we have a one-to-many relationship between a given molecular graph and the SMILES we can actually create from it. So there have been a lot of research into how you could secure a one-to-one relationship. So there's something called the canonical SMILES, where you have defined rules for what paths to take through the SMILES and in what order to branch out. But if you don't follow these rules, you can actually create a lot of different SMILES, and that will work as data augmentations. We present a different SMILES form for the neural network during training, and then it becomes more robust towards variations in the SMILES form, and learn to recognize the molecular features in different environments, so to speak, of forms of the SMILES. So that works to regularize the networks and create better predictions. One thing that's very interesting, and that's where the design comes in, is that we can train these networks to predict the next character of the SMILES string. So we usually prepend the SMILES string with a little start character or start token, and then we ask it to predict the first character of the smile. And then we input this smile, this first character, and ask it to predict the next one. So it's auto-regressive. And then after we have trained it on that, when we put in the start character, we get a probability distribution of characters out. And then we can sample this from this probability distribution. And we can say, yes, you were right. And then you get the next probability distribution. But depending on what you sampled, that will, of course, look different. And when you get to the end of the smiles, you have actually created a brand new smile that follows the smile syntax. So let's have a look here at an example. Here's the first probability distributions to the left, where we see that carbon is most dark green. So that's the most probable one, closely followed by oxygen. And some of the other characters are also possible. So let's see, we sample and it selects oxygen, and we can update the molecule here on the right-hand side with an oxygen. And then we say, now what is the next probability distribution if the first was an oxygen? And we get this one, and we sample and we start, and this is a double bond, and we can update on the molecule and so forth, and we go through. So depending on what you sample, you will get new possibilities, and that actually in the end leads to a lot of the different possibilities for generating smile strings. So it even learns long-term relationships like ending the ring before it samples the end character. And you can also see here in the middle where it comes to the 3-fluoromethyl group, it's pretty certain about what it does, because this is a very common group to find in drug-like molecules. So one of the interesting things is that if you train on a given sample of molecules, you end up with generated molecules that closely follow the properties of the molecules you have trained on. So here I follow a couple of different properties on the molecules. For two different data sets, one fragment-like and one drug-like from the Zinc database. And you can see the dashed curves are the generated ones, and they closely follow the other ones. So that's one thing we can actually mimic our known molecules, and we're interested in drug-like molecules, and we can learn these kind of distributions without programming anything about how many hydrogen bond donors do you need on average to have in our molecules. A natural question then also to ask is, do we actually generate the complete chemical space? Do we generate all drug-like molecules when we have trained on a little subset of potential drug-like molecules? So we tested this out using a generated database from Gilles Ramon's group in Bern, where he simply takes chemical rules, how can you combine things into stable molecules, and then he generates everything up to 13 atoms. And that's 975 million molecules we end up with in this database. So we take out a little sample of this. It's still 0.1%, it's still 1 million molecules. And we train our generative models, and then we can sample that and check up on how much would we actually have recreated if we sampled infinitely from it. It's sampling with replacements, so we simply need to compare it to the ideal model that have an equiprobable chance of sampling any molecule in the database. And with the data augmentation trick I talked about before, we actually end up getting models that have very high validity. It's very good at generating smiles, and it also have a very high completeness at 95% of the original database that we took the sample from can be recreated because it has learned the chemical rules there. So if we compare to some of the more traditional ways we have been handling this is via databases and enumerating compounds via fragments. You can combine them in different ways, and you can actually generate very large libraries of molecules like this. So if you have a really big database or enumerated virtual library, maybe you are on the order of 100 million molecules that we can somewhat easily handle. 1 billion starts to get problematic. So I have said maybe we can handle up to 10 to the power of 12 molecules. And that's a huge number. But drug-like chemical space has been estimated to be between 10 to the power 30 and 10 to the power 60 possible molecules, if we look at the normal size of drugs and what kind of atoms we can have there. So if we have a generative model, we suddenly can start to generate in practically unlimited chemical space, depending on what you have trained on. And to put this into perspective, I mean, if you take 10 to the power 12 hydrogen atoms, yeah, sorry, I'm a chemist, then we have 0.0017 nanograms of hydrogen. Yeah, it's so little you cannot even see it. But if you take 10 to the power 57 hydrogen atoms, then you have the hydrogen content in the sun. So these numbers are on totally different scales. So, I mean, we cannot get our hands down. We turned the usual drug finding problem, finding the needle in the haystack, into a problem of finding a hydrogen atom in the sun. But there are ways we can actually bias our network toward the compounds we're interested in. We limit the possibility. There are ways we can steer it into the right molecules. And we have over the last couple of years actually developed a couple of different methods for doing that. One of the first method was transfer learning, where you first train your generator network on a big database of drug-like molecules. It learns the rules of chemistry. It learns the usual probabilities of how do a drug-like molecule look like. It learns the SMILE syntax. And then you have a very small data set of actives. And then you can fine-tune this recurrent neural network through a few more epochs, training directly on that. And it will focus onto this one, but still generate new compounds that are in vicinity and follow the properties of the actives. Another way I'll come back to is that we can directly steer the recurrent neural network. We can train it, so we input properties and we get the molecules with the properties out that we're interested in. In other parts of the drug discovery, we are interested in making smaller changes. So we have trained it on molecular pairs, where we ask it to generate a change in a property on the molecule. So we put one molecule in. We know we want to heighten the solubility a bit. Can you please make suggestions for how to do that? And we can generate molecules that have a predicted higher solubility. Another way is also to look at substitution points. We want to keep the core of the molecule the same, and then we can ask the recurrent neural network to generate various substituents on here. Reinforcement learning also work very well. Actually, that's our main horse, workhorse here in the group, is that we use reinforcement learning to steer the prior generation into the area with various scoring functions. The good thing here is we can have a prior pre-trained, and then we can user define for each project. We can define the scoring function we're interested in via various QSAR models, docking scores, fit calculations, etc. We can try and steer the generation into this. Autoencoders are also an interesting aspect. First, we train our neural network to provide a latent space that can be recreated to SMILES. And one way is then to create de novo design is to take the decoder part after training and do the optimization in the latent space and generate the compounds there. So to return to the reinforcement learning, I mean, it is a kind of conceptual in silico mini-DMTA cycle, because we have taken the design phase and put in the generative recurrent neural network here. And of course, it's in silico, so if the molecule is valid, we can directly put it to the test and test it in various of the more standard computational chemistry methods. And then the analysis part is then taken care of by the reinforcement learning that feeds back that kind of reward to the agent and starts to generate better molecules. And compared to the real world cycle, I mean, this is no problem to in a couple of hours to run 2,000 cycles of this. And then we get a lot of interesting compound out that says they enters our standard computational chemistry workflow for evaluation and synthesis. And we have open sourced this tool, so it's possible for interested people to download it and try it out to generate molecules of your own interest. Another interesting approach I mentioned on the first slide is autoencoders, but my take on it was to actually use the data augmentation strategy to not transcode from the same SMILES form to the same SMILES form, but rather try to do a lot of augmentation either for the encoder or the decoder. And then we can get an improved latent space from the autoencoder that is more chemically relevant. It was actually quite a surprise for me that decoding two augmented forms worked very well. It was actually what worked the best, going from one form of the SMILES into another one, because how should it know what kind of molecule it should generate? But it can learn it because it has this teacher's forcing during training. So we tell it the partial solution so far, and that enables it anyway to learn the proper probability distributions and enable it to generate different SMILES forms of the same molecule. We looked a lot at the latent space and tried to figure out what is in here. How can we manipulate the latent space into what kind of molecules we wanted? And that was, you could use various genetic optimization algorithms, scans, etc., to try and generate this space. But we also thought maybe we can just teach it directly. So instead of having the encoder, we use a cheminformatic toolkit called RDKit. So we use the kind of engineered properties of the molecules, and we could encode them and just train the decoder directly from these kind of molecular properties. And then it will learn that if you have a high log P and this mole weight, you should generate molecules that look more or less like this. And here we have some test runs. The red line is the properties that we target in the various parts of the graph. And the small blue dots are then the properties of the predicted properties of the molecules that were generated using this kind of setting. And some of the settings, it is more easy to follow than others, but most of the molecules are in the vicinity of the target that we want. And we can also look at it a little bit differently, because we looked at the log likelihood distribution of the output. And we can see clear differences here between the various models. If we use fingerprints, which is a very rich, information-rich format for the input to the decoder, we get very few molecules out. It gets very sure about you need these kind of fragments and this kind of size molecule, and then there are not so many solutions to that. And then if we use the physical chemical properties, we get something that is in between the fully unbiased generation and the more information rich. And if you have a perfect autoencoder, it will only give you one molecule out, and that would be the correct answer. So suddenly we have this kind of spectrum. We can actually define how well do we want to generate the compound? Do we want to make absolutely sure that this is the compound we want to make? Or do we want to be a bit more, so to speak, maybe creative? We have a bit more uncertainty in the desampling, so we create a little bit more options to evaluate later in the drug discovery project. So it was actually mentioned in the latest Gartner report that this is now on the hype curve, and it says that they're still on the way up. So 2 to 5 years, and this technique will be totally hyped. but we actually already using it for internal projects here at AstraZeneca. But we're still not done with the research into all the refinement of these methods. So the next part is, of course, we have suggested some compounds, and then we want to see how can we make it? Can we suggest something, a route? What should you mix in the laboratory and under what conditions to get this wonderful new compound that your scoring functions and your docking and everything says this is a perfect thing. So we have two different methods. I will explain a bit more about. One is template-based methods where we use explicit little reaction cores and the transformations there. And another one is a template-free method where we use deep learning. So what is a reaction template? Here on the top, we have a reaction from the database. We have mixed together reactants that react and become the new molecule and various reagents, the solvent, and other things that are needed to make it think. And then if we can actually map what atom turns into what atom in the product, we can identify what are the atoms that changed. We can see there's a methyl group here. It changed its neighbor iodine, and there's a nitrogen that suddenly have a methyl group on. So here was actually the place that the reaction took place. And we can use that information to extract this area and its nearest surrounding into a general template that could be applied, even though the rest of the molecule would look a bit different. So there is a tool for that called RDChiral. And if we take our reaction database, our full reaction database is around 70 million reactions. Even though we keep it very generic, only one atom neighbor for the reaction core, we anyway end up with several hundred thousands of potential templates to apply to a given product that could tell us something about what kind of reaction could have made this compound here. So that's a problem. We have a search tree that is a bit different from what you see in various games, but we're still using game algorithms to solve this. If we compare to example chess and Go, we have significantly more potential steps per step, so to speak. But we don't want to take so many steps because then we get very long routes and they're not very practical in the solution. So what we do was actually inspired by this AlphaGo architecture that was becoming a Go master, so to speak. Maybe 10 years before any people thought that this very complex Japanese game could be solved by AI. But what it does is it takes this Monte Carlo Tree Search algorithm that have a range of steps. So first you select, you have a growing search tree of possible moves. And for us, they represent retrosynthetic disconnections. So first you select one of the leaf nodes and you expand it. And we can test here, of course, all of them, but that would be slow. But we use a neural network to suggest what kind of templates are actually the most potential ones to use. What seems most plausible? What is strategic? And then you simulate an output from this, and we again use a neural network just to suggest disconnection for this output. And then when we get to the end, we can see, did we actually end up finding a synthetic route for compounds we can purchase or we have on shelf, and then we can backpropagate this score and do the process again. And the neural network here are really key because they limit the search breadth of the tree search to 25, and that means suddenly we only need a couple of minutes to come back with a suggestion for a retrosynthetic path. This is also an open source project from our group, So if you have needs for retrosynthetic analysis, there is a place to start here. We have not been able to, we're not allowed to release the models we train on the in-licensed data, but there is a policy there based on open source data. So, and it's also possible to retrain the neural networks if you have your own synthetic database. Another interesting technology that is appearing now in retrosynthetic prediction and synthesis prediction is what we call template-free approaches, where we use deep learning. So we simply read in the entire smile strings into a transformer neural network and ask it to, so to speak, translate from the input smiles to that could be the reactants to the output smiles that is the product. And that way, this neural translator will learn what is recognize the possible groups that can react, and it will also know what kind of new structure it needs to have in the product. So if we train it on enough reactions, it will actually start to learn these rules of synthetic chemistry. So in comparison, we don't need to take any choices about what part of the environment for this reaction is important. It should, in principle, start to learn to recognize this if it has seen enough examples. And it's actually quite accurate when we change to the transformer architecture. We get a high accuracy in the molecular prediction. And we can also use various data augmentation strategies here, but it's a little bit, you need to be a bit more clever about how you do it and find things that are more sequence similar, then you can improve performance more. So after a couple of years, we have done a lot of interesting research. We have suggested a lot of models. Suddenly we have this model with this neural network technology, and we have that model with that one. And so we suddenly have a lot of projects we need to maintain. We need to retrain on different data sets. They have been developed by various people that have been in our department over time. And there's really no transfer learning between the different models. They don't learn from each other. So is there a way we can combine all the models? And of course, this is a rhetorical question, and I will give you the answer over the coming slides. So one of the models we have been looking at is the BART model. It's close to BERT, but it's combines BERT language, masked language model with, so to speak, the GPT models. And that means we can suddenly apply this model to both sequence-to-sequence tasks, but also more discriminative tasks. So it's usually it was trained by masking the input sequence here, language, and then we asked the model to both generate and predict the original one. So that means we just need unlabeled samples to actually train this. For natural language processing, that is a good thing. You can go to Wikipedia and get a lot of sentences to train on, or we can go to our molecular database, and we actually have quite big databases with molecules. So that seems maybe to be a possible solution for our issue here. We can make one model, we can pre-train it unsupervised on unlabeled data, and then we can maybe fine-tune it afterwards for the tasks that we want. And it will have learned something about molecules, it will learn something about smiles from the pre-training task that it can then reuse in the next step. So that was at least the idea of it. So we did this, we trained a BART model on 100 million molecules from the same database. We figured out how can we do the masking. And we also thought maybe these augmentation tasks that we have previously used for heteroencoders could also be a good pre-training task, forcing the model to learn something about the molecular graph. So that way we actually have two ways. We combine the natural language inspired masking tasks that have been very popular there with a more chemically aware SMILES data augmentation task. So here are some results on the retrosynthetic task on a data set called USPTO 50K. And we can see here that the blue line is a pre-trained one. So we start to train that. And very quickly, it actually learns to solve this task. Whereas if we initialize the model randomly and then train on the same USPTO 50K task, it takes significantly longer to actually reach the same kind of performance. And the scale here is the number of epochs. How many times do we go over the training set is logarithmic here. So we end up at 500 epochs, and the randomly initialized model have still not catched up on the pre-augmented, the pre-trained one. So suddenly we have a good situation here, at least for this data set. We can train faster, and we also get a better performance. So we also tried out another sequence-to-sequence task, and we also looked at, was it the masking task, was it the augmentation, or was it a combination of that what was best? And for both our samples with synthesis prediction forward and ratio synthesis, it was a combination of the two tasks that provided the best performance. But for molecular optimization task, where we asked the model to take a molecule and induce a wanted property change, it seemed that the mask task was actually better here. The question is if we actually found the right hyperparameters for this task. There was some indications of overfitting. But anyway, even if you had used the combined one, it would still not be detrimental, but reach more or less the same performance as if you had done by training from randomly initialized variables. So for property prediction, we compared to a more classical support vector regression model based on fingerprints. And we can see that for most of the activity tasks that we tested on, pre-training was crucial for performance. We actually got a huge performance boost on the predictive task, but we were still on average not better than the classical machine learning model. They have been optimized over a long time. For the property prediction task, where we looked at lipophilicity and two other properties, we actually saw an improvement over the support vector regression model. But the question if the features and the model was actually a smart choice for what we wanted to model there. But again, we are seeing improvements by doing pre-training over just training from scratch. So we're taking this to the next level together with Nvidia on the new Cambridge 1 cluster, which have a lot of GPUs and a good data infrastructure, so we can start to train our very big models on very big data sets. And our hope is to figure out how does these models scale. If they follow the scaling we see from natural language processing, then bigger is better. So we have good hopes here, and we can pre-train it. That's the intensive part, and then we can fine-tune it with lesser resources for the task we're interested in. So in conclusion, maybe to draw some learnings from this, SMILES is a language, I would say, for molecular structures. It has a syntax, it has a grammar, and this grammar can be learned by deep learning architectures. And suddenly, I think we can read in molecules That we have done for years. We have featureized. We have all the engineered methods of describing molecules. But this method also allows us to write out molecules in a very good way. And it can really learn to generate some molecules that follow some of the distributions without us needing to code that in. Data augmentation or smile spring. If you want to work with smile springs, it's easy to test if it works. And it's highly recommended because my experience, it always gives a little bit extra percentage points. Sometimes huge improvements, sometimes smaller, but it always seems to be giving a boost there. So that gives us many possibilities for solving a lot of diverse chemical tasks, and we can really start to play around and with the generative models and make them suit our precise needs. If we go to radiosynthesis, we can solve this with a combination of game AI and neural networks. And we can also work with deep learning technologies. We are still working on improving this to the level where it's usable. These transformer architectures are very versatile, and with massive pre-training, we can quickly adapt them to other tasks with smaller data sets and improve both training time and performance of this. So we have a lot of students who have helped out with that, and a lot of team members here. And we also have a lot of external collaborators that we have worked together with over the last couple of years. So thank you for your attention. I hope maybe we can take a question or two.
Thank you so much. That was very, very nice, I must say. From a chemist, I'm nearly convinced I should leave the lab and come to the computer much more. My suggestion is the following. I will stop recording so that we can take the question separately without recording. That will be easier. If there is any question, just post them in the chat or raise your hand. That will be the easiest. I see one hand raised.